integral of phosphorylated C-phos. And you see that different cell lines, different doses, all like lie on this single curve. Okay, next topic, because this kind of overview, next topic without complicated dynamics. You know that in physics and also in engineering, many electrical circuits have different dynamics. Could be like oscillatory, which you try to get rid of, and so on. So if you consider, like, we know that usually oscillations occur if there is like some feedback, and then here is a model which I usually show to students, because in this model simple model of rough MEC-ERG, the same MAP kinase cascade, and the only variable which is here changes is the feedback strength. So there is a feedback from ERG to rough. So if there is no feedback, of course, the response is monotonic, and then there are responses which change from transient adaptive to almost perfect adaptations, and is feedback to strong damped oscillations and sustained oscillations. But if feedback you make it positive, you have bistability. What means bistability? It means the several steady states at the same parameter values, and then means depending on your initial history, you either go to one steady state, which is almost zero, or to other steady state. Now, it appears that not only complex system can exhibit very intricate dynamics, but very simple systems. So this is like a universal cycle of protein phosphorylation, dephosphorylation, which is catalyzed by kinase and phosphatase. And this is a module of almost any signaling system. And then you see that if you have kinase which is phosphorylated by its product, so like positive feedback, then you have bistability. Now, if you take another feedback here, which would be inhibition of kinase of the product, then two feedback, negative and positive, as you know, like from classic theory, it could be like relaxation oscillations, or it's also called frustrated bistability. So the system is like bistable, and at some level it changes and flips to another steady state. It appears that there are many actually signaling circuits which can produce the same robust oscillations or bistability. And here is like, I published that there are 32 feedback designs which gives you the same dynamic behavior. Now, there are real examples, and one is like SRC kinase, which is like a single protein which is phosphorylated and dephosphorylated. This is a kinetic scheme, phosphorylated and dephosphorylated by different phosphatases, so this simple system, it's like, it could be like closed, as this is a biology already closed, open, conformation open is active, and there is also autocatalytic loop here, which is showed. They indeed can produce very intricate dynamics. So you can have either bistable behavior or oscillations, or also excitable behavior, meaning that you go like around your steady state and return to steady state with a pulse, and this is similar as a neuronal pulse, and you see how it propagates in states. So the conclusion here is that biochemical signaling system actually capable of producing any intricate dynamics which we have seen in human-made devices. Now, signaling is very inhomogeneous, meaning that it occurs in 3D, in the space. And this is why there is a title of 4D molecular communications, because you add to the time when you try to understand your dynamic system, you add the space. And why space is added. So in this classic example, you usually have a receptor on the cell membrane. And receptor is a kinase which phosphorylates some ligands, and then, some adaptive proteins or some target, and then this target moves to the nucleus, and while it moves in the cell, it's dephosphorylated by phosphatase. So again, simple cycle, but now in space. And what you see here is like 20 years old work, when we found that there should be extremely steep gradients of phosphorylation form from the cell membrane towards cell interior and the nucleus. And this is a steady state solution which is analytical for the case of linear phosphatase. And how then can we propagate signals if the signals decay so rapidly? And this is why you see these cascades. So here is again a cartoon of MAP kinase cascade. When you see that signal propagates from first level to the second level to the ERK level, and when you propagate the signal, you mitigate this decay. So the first level decay like this, the second level make like this, and this level already can propagate the signal. You can, for the simple model, you can actually found exact condition analytically for propagation of waves, and this is here. But the whole taken message is that cascades basically mitigate this effect of rapid decay in a phosphorylation signal. Here's another example of SARC where you see like all these waves, and could be like spiral wave, or from oscillatory sources could be like waves like that. So, and we think that the same is actually happening in the cell, and there are some experimental data which support that. And the next slide shows that if signals should go for really long distance, because in giraffe you have activation at this axon terminal, which is here, and then the signal, it's one neuron with so long axon should go somewhere to the brain, and then there is no way to propagate signal as I showed, and this would take many days. But because of bistability in the signal, you might have a trigger wave, and then the trigger wave actually goes, and this propagates the signal for reasonable time. Okay, now again, real example. So this is cell migration. So cell moves, and this is one of the fundamental properties of cell motility and migration. And actually, Andrei Levchenko, whom I substitute, made a great contribution to this topic. So how cell migration is controlled? There is again biochemical signaling network which controls cell migration with two GTPases. GTPases are small proteins which switch between active state when they just bound to GTP, and inactive state when they bound to GDP. And this transition, again, like in case of phosphatase and kinase and phosphatase, there are two pairs of proteins, which is called GEF and GAPs, which do this transition. So there are a network of RAC and ROA. There are two small GTPases, and there are internally mutual inhibition between them, and this somehow regulates cellular motility. So we did a simple model of this system, and what this model includes, it includes transition of RAC and RO, and also it includes RAC effector, which is called PAC kinase, and this PAC kinase inhibit GEF for ROA, and this is like why you have this inhibitory relationship, and then ROA through ROC, through its effector, activates GAP and inhibits also GEF for ROC, and therefore you have the second inhibitory, double inhibition. And we know that double inhibition, dual inhibition is actually the same as a positive feedback, and it should give you bistability. But in order to experimentally found this, we need to perturb the system. So what we do, we give an inhibitor to this kinase and see what happens. So the model gives you like hysteresis, which is a hallmark of any bistable system. And this, you see that you switch up and switch down at different positions, and this is like really hallmark of the system. Therefore, we actually introduce this how to find bistability by measuring hysteresis. And this is slides which illustrate it. So you do some perturbations, in this case PAC inhibitor, and on some level you should see a switch. And in this case, the switch is like raw GTPA level goes up. Then you fix the system here, and then, and then you go in opposite direction. Now you decrease the PAC inhibitor, and then when you do this, you should go by a different path. So if you find the same path up, like your way up and your way down is different, means the system exhibits hysteresis. So we did the same here, and indeed you see difference in blue and red. Blue is you give PAC inhibitor, and red is you wash out and then give it again. And you see that raw, which is at the upper panel, goes first up and then down, and RAC, which is inhibitory relations, goes first down and then up. So we've proved that there is a bistability of the system. But what does this mean for cell physiology? And it appears that this is the first case when we found that cell physiology is also bistable. And there is a classic assay which is called wound healing assay, when you basically scratch in the cell culture line, and then you see how a line is filled in. And then again you see the same picture. And if you do, this is like simply Brownian, like Brownian motion, random cell migration, if you do and determine the speed, average cell speed, you again see hysteresis here. So the home taking message and conclusions from this part is that basically this biochemical system determines different either mesenchymal or amoeboid movement, which corresponds to different levels of RAC and RAW. And this is very important for cancer metastasis. And a friend of mine, Edelstein Kesha, she is a pure mathematician. She just wrote a nice overview of this paper, and she showed that how you go from low RAC, high RAW, which is amoeboid movement, to high RAC and low RAW in this hysteresis manner. Okay, now the last part of the talk is something which is practical and refers to cancer biology. So again, MAP kinase pathway. Why MAP kinase pathway is everywhere? Because it's actually the most pathway which is changed in cancer together with receptors. So receptor change in cancer, like breast cancer, and Herceptin is a HER2 receptor, which is overexpressed, and then RAF, MEK, and ERK, they all might have mutations, and there are inhibitors for all of this. So the main problem in cancer treatment is that whatever treatment you give, patients will develop resistance inevitably. And then, unfortunately, the effect is that patient dies. So what I will talk about here? I will talk about BRAF mutations, so mutations in the first level of this MAP kinase cascade, and then why this mutation occurs. So, why resistance to inhibitors which inhibit BRAF occurs. So these BRAF activation mutations are found in many cancers, and these cancers are resistant to specific BRAF inhibitors. Moreover, these BRAF inhibitors cause so-called paradoxical activation. So you give an inhibitor, and instead of inhibiting the whole pathway, means ERK, you see activation of ERK signaling, and this is called paradoxical activation. And then what was found that this ERK activation is related to BRAF homoheterodymerization. So it means that this BRAF kinase can make a dimer. And the question again arises, which we addressed by physics-based modeling, is why does BRAF kinase dimerization conveys a drug resistance, and whether we can deal with it. So dimerization is part of physiological RAF activation, and what is important, the dimer have much higher activity than a monomer. And this is a feature of many kinases. RAF inhibitors facilitate RAF dimerization. This is the second, this is the reason why you see paradoxical activation. And then drug-bound protomer, which is like shown here in this kinase, it transactivates this protomer, and therefore half-inhibited dimers are active. But the question arises, why the second molecule of inhibitor doesn't bind here and doesn't stop the dimer from activity? And it appears that actually thermodynamics can answer these questions. This is again a kinetic scheme. And this kinetic scheme has cycles. You see this easily that there are cycles in kinetic scheme. For example, this RIP means kinase inhibitor kinase. You can got by reactions, starting by reactions 1 and 3, or you can got it by reactions 2 and 4, meaning that there is cycle. And in cyclic processes, the free energy change is 0, as we know from thermodynamics, and therefore the behavior of this scheme is described by thermodynamic factors. This scheme is simple. There are only two thermodynamic factors, which we call F and G. And what they showed you why you have much more kinase accumulation, half-inhibited kinase RIR accumulation from the beginning, and why the second molecule of inhibitor doesn't bind to dimer, because D2 is proportional to this term, F is much less than 1, and therefore the second molecule of inhibitor binds much worse to the dimer than the first molecule of inhibitor. You do simulations, and you see that instead of your normal inhibition curve, when you inhibit some pathway, you just go down, you have this paradoxical activation and very low inhibition. And then I thought that maybe then two inhibitors, which would have different structures, if you give here two different inhibitors, can't prevent that. And indeed, it was the case, and this is like purely thermodynamic models, which is not dynamic, it's just steady state. So basically, it's absolute equilibrium, each state is at equilibrium. But you see here that whereas neither inhibitor 1 nor inhibitor 2 can inhibit the system, if you give I2 at some level of I1, which is IC50, means 50% of inhibition, you see much better effect. And this is illustrated by this plot. So this plot is called Lewis isoballs. So these are basically levels of constant inhibition. And if they are just like concave, like here, you see that it's better than you make a straight line, meaning that the sum of two inhibitors which would have the same effect, here you have much lower dose at the same effect. So this was purely theoretical prediction, but now we are working on a model which actually puts all structural and other details in it and do some experiments with quite promising result. So this is a cartoon which basically shows you that there are two monomers which can bind to each other and then inhibitor induce dimerization. But if you combine two inhibitors, there is synergistic activation. And with this I would like to finish. And I'd like to thank people at Systems Biology Ireland. So Alexei does this model of inhibition of rough dimers. This is like a very complicated model now. It's more than 20,000 equations because it uses some rule-based called modeling. And then Horzan Fahime, she does experiments, and other people in the lab, and these are my collaborators. So thank you, and if you have some question, I am happy to answer.
Yes, thank you very much, for a very stimulating presentation. Unless I'm missing something, can you highlight what models you have used in order to study the signaling, of all pathways? Isn't just cell cultures through-dimensional, or have you gone ahead, to more organized models? Because by default, signaling would definitely change through other kind of stimuli in a more advanced model than a simple, cell bilayer.
So if you are interested, if you think that the system is homogeneous in space, then you use what's called ordinary differential equations. And then all the dynamics, like in physics, is already in this equation. So you just simply describe how the change depends on what is going with system evolution. And indeed, like people, when I started in the 90s, people didn't believe that you can model the signaling, and they asked all the questions, and first reviewers were just telling that no, it's impossible to model signaling, it always changes. But basically, ODE, ordinary differential equations, they take care about all those changes. But if you're interested in spatial propagation and in space, because the cell is extremely heterogeneous, I mean, there is no homogeneity in the cell, then you use PDEs. And PDEs is much more complicated because you need to give coordinate. If your coordinate of, say, kinases and phosphatases are not the variables, you have to give it like the boundary conditions, or just like within the equations. So it's basically, I mean, all equations are published, and you can look into it, but the whole message that, yes, signaling can be modeled very precisely.
Thank you very much. Very, very interesting talk.
Thank you.
Survey of the topic. If I understood well, your models are deterministic, so you capture the structural dynamics, both in the first section and the second section. So the question would be, how would molecular noise would affect the exhibiting either transient or sustained response? And for the second section, how would molecular noise affect this oscillatory complex dynamics?
Yeah, this is a very good question because it really does. It affects molecular noise. And I just briefly mentioned that when you do this modular response analysis, means reverse engineering, noise basically destroys all your bright theory, and you need to do statistics instead of ODEs and solving ODEs. But with noise, if you have a signaling system and some protein is like more than 10,000 copies, then it's mostly not intrinsic noise which occurs from Brownian motion, but it's noise in your measurements. But you are right, say for gene circuits, if we model here gene circuits, but I didn't talk about this, you need to write stochastic differential equations. This is absolutely correct, because you have only 1 or 2 copies.
No, very related to that, and based on your answer, instead of using state equations, would it make sense to use the Markov chain formalisms?
I never used it except of this Bayesian inference when you use Monte Carlo Markov chain algorithm to find to find like the posterior distribution. But whether we can use Markov chain for genetic circuits, yes, probably yes. But it means that you at each time you need to follow the distribution. If you have too many variables, this would be like distribution of many multivariable distributions would be difficult for large system. But for small system, it certainly could be done.
Thank you very much.
Hi, I was just wondering the particle numbers in general and the number of target size and how would it affect?
Number of what? Sorry.
Number of target size on the DNA for response regulator, for example. So it was very nice to have design patterns, motifs. Do you also have any information about those target size, and if the numbers of target size increase, would all these still be okay to simulate the behavior?
So I didn't talk about DNA at all in this context, because signaling is usually understood as signaling in the cytoplasm and in the nucleus, but by the proteins which already have been produced. You can see changes in this protein over the time in protein doses or concentrations, but not. We don't consider here what I told you. We don't consider DNA binding sites. Although we, at the same time, it's a very important area, and the work probably mostly of people in Caltech, and we also published a few papers on it. They consider again physical models with statistics, and they consider the probability of mRNA polymerase to bind to some of these DNA binding sites. And then again, you don't consider stochastic differential equations, but you consider simply, like in physics, you consider so-called Gibbs distributions by free energies. And this is like the current methods how in biology people describe activation of protein transcription of DNA into mRNA and polymerase. So it depends on binding sites and so on. And there are actually models. So if you have a DNA, you have, some gene, you have multiple transcription factors. And the equations which describe the probability of binding of, say, a set of transcription factors, because each combination of transcription factors on a molecule will cause some different effect. And there are models which, so we also publish this. Is basically, it's like physics and biology so description of this event.
Okay, so I want to ask you, what do you think about using discrete models like particle or lattice gas, accelerometer automata, models in modeling the four-dimensional signaling in the cells, especially considering that they can easily accommodate probabilistics, events and stochasticity?
I think they are useful if you have many variables in the system. In the model which I described with partial differential equations, we had like a few variables. In this case, we don't need particle-based models, because in particle-based models, you have like this arbitrary defined rules. And this is something which can be true or cannot be true, you never know. Because this is like how you postulate, you make like Markov chain of the transitions of each particle after some interactions or something. But if you write PDE, you still use physicochemical knowledge, so each reaction is like a reaction diffusion system. It goes like back to Turing, to classic work of Turing. But I think that because there is a limit of what you can do in terms of the number of variables, the particle-based approach is very valuable, but not for a single, not for two molecules, not for two different kinds of molecules. If you have more variables, it should be used. Okay, if there is no more questions, thank you for your attention.
Thank you. So I'd like to thank Professor Polovenko for his inspiring talk and keynote speech. So thank you very much for joining us again. So now we'll break for a coffee break. So we have about a half an hour. There's coffee and pastries and biscuits just outside here. What we'll do is we'll return here then at about 11 o'clock. So we have a very interesting practical demonstration with microfluidics. So we'll rearrange the chairs and we'll have, it won't be presentation based, but there may be some presentation material, but Marilla will have a very interesting practical demo for whoever's interested in attending. Okay, that's a 2-hour demonstration. Thank you.